All right, it is noon, so we'll go ahead and get started here. Rileigh, if that's all right with you.
Yeah, I'm ready to go.
All right. Well, thanks for joining us. This is our last Dairy Goat webinar for the 2025 webinar series. My name is Jennifer Bentley. I work as a dairy field specialist with Iowa State University Extension and Outreach, and glad you're joining us here today as we wrap up our series here, talking about dairy goat milk quality. And this is going to be a special focus on the Southeastern United States. So glad to have this opportunity to visit with Rileigh here today. And as we conclude our series here, I just want to say stay tuned for upcoming announcements of our 2026 webinar series. We'll have those topics out probably shortly after the holidays. We'll get those put out. So I'll send out a email blast to the participants that have registered for our webinar series in the past. As a reminder, this is part of our Sustainable Dairy Go Agri-Systems grant project that we're under with the National Institute of Agriculture, Food and Agriculture. And so if you want to learn more about the project, you can go to www.dairygoagrisystems.org and learn more about what our project entails, as there's a lot of working parts going on behind the scenes, and we hope to bring more of that information and research to you in 2026 as well. So go ahead and get started here today. I'd like to introduce Rileigh Powers. She is a graduate research assistant, obtaining her master's in animal science at the University of Tennessee, Knoxville, under Dr. Liz Eckelkamp. Her research projects focus on small ruminant dairy management and milk quality. She obtained her bachelor's in animal science from the University of Wisconsin River Falls in 2023, during which she worked at the University Dairy Farm and performed undergraduate research. She grew up on a small dairy community in South Central Wisconsin and worked on her family's goat dairy, which is where her passion for the industry ignited. And she's an active member of the American Dairy Science Association and coaches the Dairy Challenge team at University of Tennessee. So welcome, Rileigh, and we look forward to your presentation here.
Thank you so much. I'm super excited to be here today and talk through my research. But before I get into the nuts and bolts of my research, I want to go over a little bit of background. So the dairy goat industry in the United States is actually about 15 years behind the dairy cattle industry, especially when we're looking at technology, artificial insemination, and even support from our industry professionals. In fact, goats only have about 15 drugs labeled for use, and none of those are labeled for use in lactating animals. And this is really an interesting point to me because worldwide, dairy goat populations, along with dairy cattle populations, are still rising. And when we focus back in at the United States, production, population, and yield is rising. And it's just super interesting to me that we're having this lack of information available, especially when, as milk yield rises, we're going to be worried about our milk quality. And the first thing that we think of when we hear the word milk quality is going to be somatic cells, bacteria, and maybe a few other things. Looking a little bit more at our somatic cell count, which is a little bit confusing at times, somatic cells are just white blood cells that are circulating within the udder. And there's three main types that we look at when we're looking at somatic cells, and those are going to be macrophages, polymorphonuclear neutrophils, and lymphocytes. And they each have different roles, but when we're looking at the differences between cattle and goats, cattle are going to have a majority of macrophages, which those we're going to see being our number one also in humans, but goats are a little bit different. They have polymorphonuclear neutrophils as their main cell type. And the reason why I'm pointing this out is because in cattle, when they have mastitis events, polymorphonuclear neutrophils are going to be their main cell type. Whereas goats, that's already their main cell type, and it actually decreases in percentage of cells when they have mastitis. And these differences between species are a little bit exacerbated when we look at the secretion type of these animal species. So dairy cattle are going to have a miracrine type secretion. And that's just where, when those milk fat particles are secreted, they're just going to be pushed out of the cell. No cell damage whatsoever. But in goats, they have an apocrine type secretion. So when they create those milk fat particles. They're going to be pinched off within the cell, and that's going to have a little bit of cellular material or cytoplasmic particles within that little bubble of milk fat particles. So when we're looking at somatic cells underneath a microscope or even through the machines that we use, there's a little bit of noise when we're looking at it, because those extra particles are going to be about the same size as the somatic cells that we're looking at. So instead of counting only somatic cells, which is what is used in dairy cattle for looking at milk quality, we're getting higher numbers than we really should. But to kind of bring it back into some similarities, goats and cattle always increase as lactation progresses and as days in milk increases. But what's a little bit different, as we all know, is that dairy goats are going to be a little bit higher than dairy cattle. At the end of a dairy cow's lactation, their somatic cells are going to be about, in their later lactation, 300,000. But dairy goats are going to kind of start more around that level. Something we're all concerned about when we're looking at selling fluid milk is going to be our limits, our governmental limits. For cows in the United States, it's about 750,000, but for goats, it's at 1.5 million, trying to allow for that higher level that goats consistently have. But what I found really interesting when doing some research was that the European Union has their cow limit set to only 400,000, but there's not a limit set for dairy goats. And this is supported by research in that it's all over the place. But Souza et al. Did a lit review looking at all these different papers about somatic cells in dairy goats, looking at healthy udders and those that had mastitis. And between and within studies, we're seeing overlap between those ranges, where some of the animals in the pathogen or mastitis category are going to have a lower somatic cell range that is overlapping with our uninfected. And I would really like to point out that none of these papers, even though this research study was done in 2012, had any representation from the United States. Going back to our milk quality, we were kind of talking about somatic cells, but what about the bacteria? If we're going to have mastitis, we're going to be concerned about what kind of bacteria is in there. So looking at the pathogens in relation to somatic cell count level in dairy goat milk, it's a really busy table, but bear with me. Over almost 50% of our pathogen free is going to be within less than that 1 million range. But also 13% of our minor pathogens are still in that lower range. Same with our minor, and our major are about equal between our high and low categories. So what this table is telling me is that, while yes, a majority of our pathogen-free samples are coming from that lower range, we're still having a lot of those major pathogens in our infected udder halves being within that lower range as well. So there's not really a big distinction between our less than 1 million and our greater than 2 million. An overview of kind of some of the bacteria that we're mostly concerned about when we're looking at mastitis is going to be our Staphylococcus, Streptococcus, E. coli, and Mycoplasma. And we look at them a little bit differently. We look at them a little bit differently. So Staphylococcus aureus and non-aureus staph, we're going to treat differently, even though they're the same species. And that's because one of them is going to be infectious. So Staph aureus, when one animal gets it, depending on our quality protocols, we might see that spread from animal to animal or animal to us. So if we have a cut and we get staph in it, we are also going to see that within, we could get infected with that as well. Streptococcus is the same thing, where we're gonna see it both contagious and in the environment. So the environment's gonna be our bedding, our milking machinery, or even our hands. E. coli is going to be mostly from the environment. So that's going to come a lot of times from fecal matter. And then mycoplasma, we're really only going to see that if it is contagious. And this matters because when it comes to treatment, so like I had said earlier, there's only 15 drugs labeled for use in goats, and none of them are used in lactating. So what are we going to do when it comes to mastitis treatment when we have all these different bacteria that need different treatments because they're completely different species or they come from different areas? A little bit more on looking at milk quality factors. There's animal factors. So our age, parity, stage of lactation, breed by breed differences have been very well proven, as well as our environment, which is something we do think about a lot, looking at teat disinfecting, our bedding, under or over milking and trauma to the udder or teat. So if we're seeing severe over milking, we can see that causing issues with our teat end, which our teat end is what's going to be protecting that udder from the environment. Another thing that we tend to kind of forget about is gloves. So Staph aureus is going to live in our hands. We naturally have that all over. So if we're not wearing gloves or not cleaning our hands properly, that's another way that we can affect our milk quality without even quite realizing it. So milk quality, somatic cells, the environment, everything, the bacteria that we're looking at, all this confusion and this disparity between the standards that we use being compared to dairy cattle and the lack of treatments available brings me to the question of what's going on in the US, and more specifically, what's going on in the Southeast. The Southeast is an under-researched area of the US because there is a smaller proportion of dairy animals in this area, so it's not always the main focus of a lot of research trials. So what's going on down here? My first study is a survey that I did. My hypothesis was that adhering to best management practices would be associated with a lower somatic cell count and a higher milk yield, with the objective to understand current management practices in the US, focusing on that small southeastern region. Now, I distribute it through Qualtrics, focusing on the US, and then I distributed that online Qualtrics survey through social media, email listservs, word of mouth, kind of however we could best get it to people. And then mailers, I focused on the Midwest only, and that's because a majority of our dairy animal population is focused in that area, and the high proportion of individuals who may not have access to the internet in that region. So that's why they were mailed to the permitted addresses that I compiled from there. And the question format was based on the Tennessee Quality Milk Initiative that happened a few years ago, because they had a good success with understanding the region's milk quality. So there was 11 open-ended questions, 50 multiple choice, 13 Likert scale blocks, which had about 53 questions in it. And it was a very long survey, very comprehensive. So this is an example of a general respondent information, which has, this one is one of the most important questions. Are you operating a working small ruminant dairy farm in the United States? So more farm specific would be, how long is your doe or ewe's typical lactation, especially when I'm thinking of European dairy goat facilities. Some of those do employ extended lactations, which is when they do not dry off their does at the end of the lactation and they keep them milking for sometimes 2 years, sometimes even more than that. And then asking some more milk quality focused questions, such as how do you test for mastitis? The online results, I had 24 responses, 5 of which are in progress and 12 completed. The online did not do nearly as well as the mailers did. So I sent out 750 mail-in copies, 4 of which were returned for wrong address. I got a total of 107 responses, 66 were completed. That gave me a 14% response rate, with over half of those responses completed, giving me an 8.8% overall survey completion rate, which doesn't sound like a lot, but in survey work, that's actually a pretty good number. For data analysis, everything's still ongoing. Unfortunately, that's doing hard copy survey input is one of the first things to kind of go when life gets busy. So that's been put on the back burner a little bit. But some general preliminary results that I've seen is that the mail-in copy was much preferred for the online. Hypothesizing, probably because it's a lot easier to return to a paper copy than it is to an online copy. I know that if I do anything, reading papers especially, I like to print it off and then read through it and go through it. I am not someone who likes to do online, so I'm thinking that might be it. Reminder that this is very preliminary basic results. So the herd size that I saw from a glance would be a range from anywhere from 2 animals to 700. And it seems that herd size overall from these survey responses is increasing. And then a lot of things that we do here at Tennessee in our dairy lab is looking at value-added agriculture. So on-farm cheese making is a really good example of that. On a scale from 1 to not at all to 5, very likely, it didn't seem that a lot of people were looking at value-added agriculture as being something they're going to employ in the next 5 or so years. But when looking at continuing farming, increasing net farm income, reducing antibiotic residues, improving milk quality, and increasing milk production, it seemed like a majority of the respondents were strongly agreeing with those statements, which is really, that makes me feel good, especially since my dairy goat farm back home, we had to stop milking. Life just got to be too much and we couldn't keep milking. So it's great that people are wanting to continue farming as a way of life. And not many people are seeing mastitis persisting despite their efforts to control it, which is great that even though we don't really have any antibiotics that are prescribed for mastitis in lactating dairy goats, that we're able to control it. My study 2 is going to be my big study, and it just finished this week, so I'm really into the data analysis now. And this was focusing on Tennessee goat dairies. My hypothesis was that decreased somatic and bacterial cell counts would be associated with lower parity, so lower amount of kids that the goat has given birth to, improved milk components, and decreased cases of clinical mastitis. And my objective is just to develop benchmarks, see what is going on in these Tennessee goat dairies. So for my study design, I looked at Tennessee permitted goat dairies. There were 6 at the start of the study, 3 of which enrolled in the study. So I get to say that I got 50% of the Tennessee's dairy goats enrolled in my study. Rolling enrollment began this spring of 2025. And the way I did that was as long as 5 animals had kidded and were 3 days post-kidding to be out of that colostrum. And 95 total animals were enrolled in the study. Looking at my animal health portion, I did quite a lot of animal health diagnostics. So looking at hygiene, hocks and knee scores, ocular nasal discharge, locomotion score, body condition score, heat stress, kidding score, and mastitis score, of course. But those don't really mean anything unless I describe a little bit more of what I'm seeing when I'm looking at these regions. So for hygiene, I looked at lower leg, upper leg and flank, and udder. And that was on a 1 to 4 scale, with 1 being free of dirt or very little dirt. So I think of those as kind of our show animals, especially when I'm thinking of dairy cattle. Those are going to be cows that are cleaned and ready for the show. 2, slightly dirty. They've just been, living life. 3, they're going to have some mud on them, but they're not going to be completely covered. And a 4 is going to be, they've been rolling around in the dirt pretty good. When I'm looking at hygiene, lower leg is going to tell me a lot about where they're walking. If they're walking through a muddy pasture, or if their bedding is really clean, it's going to tell me based on their lower leg. For upper leg and flank, that's going to tell me a lot about where they're laying down. And when an animal is laying down, we're going to think about their udder being in that same area. So those 2 are going to be a little bit more correlated, where if their bedding is dirty or there's always one that decides to lay in a mud puddle, we're going to be able to tell that from these hygiene scores. For animal health, hocks and knee scores, Hocks are going to tell us a lot about when they're laying down or where they're walking. Um, and then knee scores, goats tend to, when they're grazing, especially on smaller, shorter grass, they're going to be on their knees a little bit more. So one is going to be, there's hair on it. Two, lesion or a scab. And I saw a lot of twos, especially on knees, because they are climbing up on stuff, putting their knees on everything. So there's a lot of lack of hair, but not necessarily lesions or scabbing. And then three is going to be a for sure injury on that area. Ocular and nasal discharge, just whether or not they seem sick. 0, no discharge. 1, they had a little bit of discharge. And then locomotion. I did it on just a 1 to 3 scale to make it really simple and easy to do during milking procedures. So 1, they're sound, or maybe they just walk a little funny. 2 is going to be they're limping, especially if they have a little bit of an arched spine. 3, they're going to be severely lame. They're not going to want to walk at a normal pace or even get up at all. Body condition score. 1 is going to be, that's going to be a severely emaciated animal. We're going to see the backbone, the ribs, they're going to be quite hollowed out. 2, we're going to see the backbone and some ribs, but they're still going to have a little bit of meat on their bones. They're not overly emaciated, especially considering that we are working with dairy animals. We're going to see that 2 to 3 range, which a 3 is going to be, we're not really going to see the backbone. We're going to maybe see a little bit of the ribs, but they're going to have some fat over their body. And then 4, those are going to be our chunkier animals. We're not really going to be able to see that backbone or ribs because of a thick layer of fat. Looking at the heat stress portion that I looked at, I used a hobo monitor, which is just a simple little contraption that I can put up in the top of a barn. And I offloaded that every single visit. And then I used THI benchmarks, which aren't thoroughly proven for every breed of dairy goats because every breed is so unique. But the general benchmarks would be THI, temperature humidity index. When we average those out, a 75 to 79 would be a little bit of heat stress, but we're not really going to see that in our healthy animals. And then a greater than 90 would be extreme. We're going to see panting. We're going to see a drop of production. This is a little bit of a nicer way to look at it, where if we're seeing a temperature of, let's say 80, once we get up into the 60s and above for humidity, we're going to see some of that potential heat stress. But once we get into the mid 90s, if it's super humid, which it does like to get in Tennessee, we're going to see a little bit more of that moderate heat stress and maybe see some effects on our milk production. Animal health with our kidding scale, kidding and amount of kids born, kidding scores, and let's see how many kids were born alive have been shown in other studies to affect milk production. So I wanted to look at this as well. 0, no problem, gave birth without assistance, or you didn't even know that she gave birth at all. 1, there might have been a slight problem, we checked. 2, they may have needed a little bit of assistance. You just kind of pulled the leg, just corrected the head position. And then 3, they had some considerable assistance. We had to deliver manually. And then 4 would be, we had to call the vet out. It was a very difficult birth for that animal. Let me go back quick. This is actually a picture from our goat dairy when we had it, of one doe who had 3 kids, 2 of which were from one of our bucks and 1 of which wasn't. I actually scolded my sister when I walked in and took this picture because I was like, whose baby is that? Like, did you move one of the does? And she was like, I didn't move any of them. She was the only one in that pen. And I searched for probably a good hour looking for a doe who had just given birth to have it be only her. So that was a really fun time. And then mastitis score. I've been talking a lot about milk quality, so I should probably look at mastitis scores. 0, no milk changes, looks like perfectly normal milk. 1, we're going to see those clots and flakes in the milk. 2, we're going to see those clots and flakes, plus maybe that udder's a little hot, or you feel a little bit of a hard nodule. And then 3, looking at an animal who is fully sick along with those milk changes and udder changes. This image is not of a mastitis milk, but it is of a really unique sample that happened. I had taken this sample, transported back here to UT, and after refrigeration, it separated, even though it was perfectly normal milk. So that was really, really interesting for me. Now, my true milk quality design, I had to look at the milking itself. So for milk yield, I use the Waikato milk meter, and that gives me range from 0.5 to 10 pounds in quarter pound increments. And with that, I was able to get individual composite samples. So basically, I got a sample from both udder halves instead of taking samples at the beginning of milking or post milking. Mastitis samples, they were taken from suspected animals at the quarter level. There was not a lot of suspected animals, especially just based on visual changes, but we also used the composite samples as a way to investigate suspected animals based on bacteria counts or somatic cell counts. And we did that at the quarter level because once you do a composite sample, it's really hard to tell which udder half is infected with mastitis. And then bulk tank, I took a bulk tank sample just like the state would, or just like you would if you were looking at it using just a dipper and using the same stabilizer pellet that I did in the individual samples. So for analyzing these samples, I came back here to UTIA, where we have a Center for Dairy Advancement and Sustainability, Precision Dairy Quality Laboratory. We have a few different machines that we use. The FOSS Milk-A-Scan is one. We also use the Bentley occasionally, and looking at those two, we saw somatic cell counts and then bacteria. We did two different counting methods. Colony forming units, which is an approximation that we use when we're also culturing bacteria, so making it grow before we count it, and then individual bacteria counts, which is going to try to estimate how many individual bacteria cells are there. For milk components, that's where we use the Milk-A-Scan, and we looked at fat, protein, total solids, and more. There's actually quite a lot that are not listed, but I did not include those within my study because they're not validated by the parent company for use in dairy goats. For mastitis samples and somatic cell count differentiation, I used the Minnesota Easy Culture for clinical mastitis to look at mastitis samples. That is a really quick and easy way to see what kind of bacteria we're having in those samples. So I can look at gram positive versus gram negative, and then like staph streps in order to kind of give us a little bit of a better idea as to what we're dealing with without having to do a full bacterial culture, serial dilution, and all that extra stuff that I didn't think I had time for. For somatic cell count differentiation, I chose 6 animals from the first 2 farms enrolled, 2 high somatic cell dose, 2 moderate, and then 2 low based on the farm mean from the first 2 samplings. So after the first 2 samplings were done, I looked at the average individual somatic cell count, and that was how I chose some indicator animals, because the somatic cell count differentiation takes me about 30 minutes per 6 samples. So if I were to have done a herd of 60, that would have taken me 5 hours, probably more than that, because I would have had to take a break at some point. So I just chose indicator animals to kind of give us a little bit of an idea as to what are those different cell types in the milk that I was talking about earlier. So with all the crazy stuff that I just mentioned, this is my study layout. So I did all the scores, collected all the samples on that week 1, which I also called my individual visit. On week 2, I got a bye week. So that's when I did all my lab work, did reports for the farms that were in the study. And then week 3, I considered my bulk week. So I took off locomotion and body condition scores because those change so slowly over time. And then I also took off milk yielded individual samples because typically when we're looking at test days, we're going to do a once a month test if you are on DHI test or anything like that. So we're kind of modeled it after that. That way I wasn't spending hours upon hours taking up that on-farm time. So my timeline, the first farm was enrolled in February 26th of 2025. And like I said, throughout the trial, the farmers got reports on animal health, milk data, and heat stress. That was one of the perks of participating in my trial, along with working with me. Just kidding. The end date, the last farm stopped yesterday. So I'm finally done with collection, even though I'm really sad that I don't get to go on farm anymore. Preliminary data, and this is really base. This is just for me throwing together kind of everything and seeing what might be correlated with one another. This is kind of how it comes out when I first run it through just a correlation table. And it's a lot to look through, but just to kind of describe what I'm looking at here, this tells me how highly correlated it is and if it is significant. So for protein versus casein, the closer that top number is to 1, the more correlated it is. So it's basically, they're in a 1-to-1 ratio, which makes sense because casein is a protein and we want a lot of casein within our milk, especially for making cheese. But when it's really low and that second number isn't below a 0.05, it's not really something that's going to tell us much of anything based on this study. So somatic cell count and casein didn't really correlate, which makes sense because protein is going to be produced no matter what, how many somatic cells are produced within the udder. So just some general correlations that I have seen from this preliminary data is that as milk yield increases, protein, casein, our solids, non-fat, and urea decreases, which makes sense because of dilution. So if you see that during this time of year, when we're tapering down milk production, or if you already have, as our milk yield is tapering down, we're going to see thicker, fattier milk that just has more components in it because we have less milk to dilute those components through. And then as that temperature humidity index, so as temperature and humidity rises, we are also going to see fat, protein, lactose, casein, and total solids decrease. Which that didn't really make sense to me when I was first looking at it, because when I think of high THI and heat stress, when an animal is heat stressed, I think of lower milk production. And so I would assume that there would be more fat, more protein. So that was a really interesting snippet that I saw from my preliminary data.
Rileigh, could I jump in there just real quick?
Yes.
So you're basically saying the total value, if the milk production increases, the total pounds of those is increasing or just the percent?
The percent. So as milk yield generally increases, our percentage is going to go down a little bit, not total, but that if we have an animal that produces 10 pounds of milk at the start of lactation, and she's at a 3% fat, 3% protein, just to make math easier. If she goes up to 12 pounds, we're going to see a slight decrease in those numbers because there's a limit to how much fat and protein that an animal can produce. So we are going to see those kind of correlate to where we're going to have a little bit lower production. Not that we're producing less or more protein or fat, but that we're having a little bit more milk to dilute it in a way. Does that answer your question?
Yes, very much so. Thanks.
Okay. And then I just threw this together to look at some of that health data I had gone on and on about. When looking at hygiene, as lower leg hygiene increases, so as we have poorer hygiene, we're also going to see the other hygienes increase or have lower hygiene, which makes sense. I mean, if we expect our animals to kind of be clean overall, or if it's been a really muddy year and we have pastured animals, we're going to see them all be a little bit dirtier. It's nothing bad, but that we're going to see those all correlate with one another. Because if they're walking in mud, it's going to splash up on their udder a little bit. If they're laying in mud, it's going to get on their udder. It's probably going to get on their lower legs too. So that all makes sense. And then I did a little bit of a histogram just to kind of show you what I was seeing. So remember, a 1 is going to be free of dirt, and a 4 is going to be quite a lot of dirt. So of all the farms that I looked at, overall, we're going to see those clean animals. Even during those little bit of muddier periods, we're not going to see all of our animals be completely covered with caked on mud. But this was just a nice way of visually seeing that for udder, we were keeping those udders really clean. 93.5% of all the observations that I did for a 10-month long study, that udder was clean. The lower leg was a little bit dirtier at times, especially if they're getting into the mud and playing in it like goats like to do, to be pains in the butt. But they're still not getting severely dirty. There's nothing that we were ever concerned about within hygiene. Mastitis, some of the general pathogens that I found was suspected Staphylococcus species. So at the top, we're going to see a Staph aureus, and that's indicated by that clearing around the cell. So that whole thing should be completely red. And instead we have that clearing. And hemolysis is just a fancy term for the bacteria is breaking down blood cells. And then we also saw suspected E. coli. And I say suspected because we didn't send these bacteria off for confirmation through DNA. So I cannot say absolutely that that was a Staphylococcus aureus or that that was an E. coli, but it is a suspected, it should be what it is. It should be a Staph aureus or an E. coli. And then my somatic cell differentiation, this is a glimpse as to what I see underneath the microscope. And I saw high variation so far. The cells are really hard to differentiate from one another. And when you're looking at them under the microscope, if I cannot clearly see that that is for sure a cell and that cell isn't intact, I cannot count it. So it's a little bit more difficult to differentiate by hand. So wrapping up, some implications and future directions. Finished data collection, but for further analysis, I want to look at areas of need on farm and correlations between components and health metrics. That's a little bit more of a complex method of analysis, so that's going to take me a little bit longer than just looking at health versus just looking at components, because those are two different things in the realm of statistics, and you're not allowed to look at them with each other without a little bit more complex modeling. And then some potential future directions. Just, I have 1,000,001 ideas, but I narrowed it down a little bit. Looking further into our microbiological analysis of mastitis. So taking more mastitis samples or taking more samples in general to look at those mastitis cases and see what kind of bacteria are we seeing and which ones are going to be the most problem for our farms. Methods of detecting subclinical mastitis. I can say that I only saw, I think, one case in this 10 months over 90 plus animals of clinical mastitis, but we're still seeing issues with some randomly increasing somatic cells or randomly high bacterial counts for one doe. So I think moving further into the direction of seeing if we can do something about the subclinical cases of mastitis, so the ones that we're not going to see visually. Antibiotic research, I think, will be really helpful. If we can see the mastitis, but we can't do anything about it, what's the point, you know? And then milk quality and cheese making is something I'm really passionate about. So looking at those sensory characteristics, the reason why, especially in dairy cattle, we are so concerned about somatic cells is once we get to a certain level, it gets super salty and it has an off flavor, and it's not enjoyable to consume at that point. So seeing at what level are we going to have those sensory characteristics change for our dairy goat milk? and what correlations should we be looking at. I went a little bit faster than I had anticipated. I'm sorry, I got really excited. But does anyone have any questions? And feel free to take down my email and reach out to me if there's anything I don't answer.
Thanks, Rileigh, for your information, and looking forward to, as you get more deep, take a deeper dive into your project results, we'll learn more about some of those correlations that were referring to. But looks like we have a whole lot of questions in here, so I don't think you have to worry about um, being done early. But Larry did ask a question early on. I think this was when you were talking about parity. Is parity defined as each kidding or number of kids? Because in like dairy cows, we're assuming parity is per calving
Yes.
without concern with multiples.
That's a good question. I should have been a little bit clearer on that. So parity is going to be each time that they kidded, not how many kids they have had. So they could have 3 kids and be in their first parity.
Okay, thanks. And then there's another question in here about this study was for permitted goat dairies. Does that mean dairies that are USDA grade A certified, or did it include goat dairies licensed by the state as pet food producers?
Yeah, so I pulled grade A and B certified dairies based on the state. So if they were permitted through the state, then I would have gotten that information.
All right. And then another health related question on ocular discharges. Were they relative to dust, poor ventilation, worms, or other things?
Mhm. That's a great question. So I did look at housing before and after study, and I was in the housing, so I did take notes on if I saw any big changes in dust or ventilation, and I didn't see any relativeness. I think I only saw, I could probably count the amount of times I saw ocular discharge on my hands throughout this study, so I didn't see any high correlation with that, especially given I did see some high helminth infections in some cases, and I didn't see much difference there on these farms.
And I had a question that I had put in here about when you were doing the mastitis scoring, were you using the CMT paddle?
Nope, I did not use the CMT paddle. I just used visual identification, taking my gloved hand or letting the farmer do that if they have a squirt cup, just using basic visual identification. I had done a lot of research on the CMT efficacy in dairy goats, and because this was more of a benchmarking research trial, I didn't want to get too in depth into looking at mastitis, especially given that the CMT, while it is good for looking at mastitis in dairy goats, it's not highly proven to be like as good as it is in dairy cattle.
All right. And there was this comment in here about a commercial dairy. This is Christine, a commercial dairy located in Western North Carolina. She's interested in participating in future studies and surveys where applicable. So I think
Awesome.
you still have your email listed here. So
Yeah.
would that be a good way of getting in contact with you about any future studies?
Yeah, that would be great. I won't have anything this year because I'm going to be focused on, unfortunately, sitting behind my desk and writing and finishing up my master's, but I'm sticking around for my PhD because I have too many ideas and I need to be able to continue doing research. So I'm going to be definitely doing some more research trials.
Great. And I would say, Christine, as part of our Sustainable Ag grant, my colleague Larry Tranel has been doing farm financial analysis on dairy goat profitability. And so that would be another way of participating in some of the studies that we're working on currently too. So you could always reach out to Larry or I on that as well. And then there's another question. Any recommendations on specific all-inclusive laboratories to use that are local to Northeast Tennessee? They have to send off to one lab for somatic cell and then specially request each individual bacteria count and then send off for another lab in Virginia for nutritional analysis.
Yeah, that's definitely tough. We do not have a lab in Northeast Tennessee for sending off, but if you would like to send me an email, we do have this lab here and it is available for use. So that's something that me and Dr. Eckelkamp can talk with you further about for getting your samples run.
Well, thanks for answering all those questions, Rileigh. And there was a comment here about getting the recording for this webinar. And so give us about a week to get this edited and up on our YouTube channel, and then I'll send out a reminder that they're all available on our YouTube channel with the link so you can further review it and the other webinars that we hosted throughout the year. As a reminder, I did also put in here the evaluation link for today's webinar. If you wouldn't mind clicking on that, that just helps us with our programming and gives us an idea of your understanding and learning of each webinar that we do, and also looking for topics for the upcoming year. So if you have any specific things that you're wanting to learn more about, we sure appreciate your input in that. You can always email us as well, and we take those topics too. But I'm kind of in the planning stages of putting the webinars together, so I would certainly take any topics of interest to our producers here. I'm not seeing any other questions, Rileigh, so I think we'll conclude here. Again, I appreciate you being on our webinar here today and look forward to seeing everybody in 2026. Thank
Thank you
you.
guys so much.